We can hear you. You're ready to start whenever.
Okay, great. Awesome. Thank you. Okay, so first of all, I'm really sorry I can't be there right now. I tore my ACL and just had surgery, so I'm on crutches still, and I figured the ice would not be great. Second of all, thanks for the invitation, and also thanks for Will for doing such an amazing job of starting off this session. I feel like I'm going to be sort of the poor man's version of what you talked about. That was really beautiful work. So today I'm going to talk a little bit about some work that was done in my group in the last year or so on developing a robust nonlinear latent manifold for some single-cell RNA sequencing data. And the flavor of what I'm going to talk about is actually quite similar to what Rahul talked about just now. And we'll talk about, oops, sorry, how do I, this isn't good. Yeah, okay, here we go. Okay, so as we know, sitting in this room, single-cell RNA sequencing opens up a whole bunch of really exciting opportunities to understand how cells work together to create biological functions in tissues and organs and whole organisms as well. So the idea really is that when we think about developing atlases or just querying RNA in single cells, we're really interested in not only understanding single cells and their states, but also how cells interact in spatial ways, how they depend on each other, what goes on between cells in the extracellular matrix, and again, how they respond to particular perturbations, maybe exposure to a drug or different kind of genetic variation as well. So there are a whole bunch of challenges, as we know, when we think about just single-cell RNA sequencing data coming off of the machines that Rahul was talking about. So in particular, as a statistician and someone who does machine learning, some of these challenges with single-cell data are quite unique to this particular technology. So first of all, as we've sort of seen a little bit already, often the cell types and cell state is poorly defined. We don't really have good ground truth. Also, cell types are not necessarily distinct, but maybe they're sort of a more continuous definition of a cell type. Something that we see a lot in these data is that there are rare or continuous or unseen cell types where we just see some kind of cell that looks like an anomaly, like an outlier. And in particular, when you look at tumor data or data from different organisms, often this comes up a lot. So batch effects, dropout, and doublet detection are all variations of sort of the technical biases of the data. And some of these are fairly well understood. In some level, we do understand a little bit about how batch is going to affect the data, specifically the expression of RNA. Similarly with dropout, dropout is not sort of a technical glitch, but it's actually just sort of a byproduct of sampling from a multinomial distribution. Similarly, doublets, we sort of understand in the drop-based single-cell RNA sequencing what's going on there, but detecting them is actually a little bit harder than it appears at the outset. And then another thing that I think actually comes up a lot in the data that we've been analyzing is this idea that there's a heterogeneous latent dimension. So what that means is that when we think about sort of the complexity of each of the samples that we have, it turns out that actually it's quite heterogeneous. So for example, if we think about a whole blood, or we think about brain, or testis, or thyroid, the complexity of the data, the complexity of just RNA sequencing is actually much, much greater in those data sets in our hands than some other cell types and tissue types that we've been analyzing. And so being able to sort of understand the different latent dimensions and let the data drive those latent dimensions, I think is incredibly important. And this sort of theme is going to come up over and over, where I feel like one of the most important things to analyze and to get right in single-cell RNA sequencing is understanding variation across the population. At the end of the day, I sort of grew up in this world of eQTLs, where we're really interested in finding genetic variants that affect gene expression. And in order to do that in these types of data sets, we need to so carefully model a variation within a population, controlling for variability among the separate cells in a single sample, and also controlling for batch, which occurs across samples. And so getting that variability, that variation right, is something that I'm extremely concerned about in these methods. So in order to think about addressing addressing all these problems, we developed a low-dimensional model of RNA sequencing to deal specifically with these issues. And the goal at the end of the day is to basically develop a platform upon which we can develop all sorts of downstream analyses. So, like I said, once we have some low-dimensional manifold, which is our goal with the work that we're talking about, and Roel also very nicely explained in the last talk, we can do so many things downstream, right? So on this latent manifold, we can think about doing normalization, where we sort of get all of our different cells on the sort of same scale. We can do batch correction, and I'm going to show you an example of how we do that later on. Which is really just the problem of manifold alignment, exactly as Rahul was talking about. We can impute missing values by basically taking new points in this lower dimensional space and mapping them up to a high dimensional space where we have those the higher dimensional values filled in. We can do pseudotime estimation, where we think about diffusion in this low dimensional space of each of these different cell types. And cell states. We can think about visualization. Obviously, in a low-dimensional space, we can visualize the distances between each of the different cells. We can do density estimation or posterior modeling the posterior distribution of this low-dimensional space. This is, I think, going to be particularly important when we start developing really sort of aggressive approaches for experimental design, where we have specific density estimates of a sample in this low dimensional space. And what we're interested is thinking about how to take that low dimensional space, the distribution on that low dimensional space, and warp it and morph it to another density by choosing cells appropriately, by choosing cell types appropriately, using something like back sorting. I think one thing that's absolutely critical in this domain, especially since we're sort of building everything on top of this method for dimension reduction, is uncertainty estimates. So a lot of the downstream analyses that I've talked about are building on this low dimensional representation where there is amount of uncertainty, but often that uncertainty is tossed away just because it's sort of not modeled properly, or it's really hard to incorporate in the downstream analyses. And one thing that I really like to do in the models that we're developing is to quantify uncertainty at a very precise way, and then propagate that uncertainty to the downstream analyses, just so we have sort of appropriate error bars on how well we're doing. And then finally, like I alluded to, my group is doing a lot of work on experimental design. And the idea there is really to think about how to choose cells, how to choose different types of experiments, or different samples, or different genotypes, genotypic backgrounds, how to select those in such a way to sort of maximize information from each one of the experiments that's being conducted, because obviously they're expensive. Okay, so that's our sort of goal. I'm going to dive into the math. If this is not interesting to you, then you can kind of space out, but I actually feel like there's some interesting signal in the equations that we can sort of talk through and get information from. So what we do to be able to build this nonlinear manifold is use something that's called a Gaussian process latent variable model, which is just a nonlinear latent manifold. So it's a nonlinear space, and there's a sort of a picture of it in this slide. And you can see that it's, again, highly nonlinear, but it's also very smooth. And what we're doing is really finding this latent dimension, this nonlinear latent space, making some assumptions about the smoothness of the latent space. And so the way that we do this is we have our variables y here, which are going to be our observations. So you can imagine this as a n by p matrix, or a cells by gene expression, gene expression matrix in the reals. We can think about it in the reals or as counts as well, but I should say that we do the log normalization, or log normalization plus 1 for each of our observations after doing other sorts of cleaning. What we're going to do, our goal is to then estimate this latent space x. So x is going to be of dimension n, so that means for every cell, we're going to have a representation of that cell in this latent space. The latent space itself is going to be of dimension q, and q is whatever we choose. So let's say for the experiments downstream, we're going to choose q equals 9 or 10, and then we're going to have 10 different dimensions upon which we're going to map each of our cells. So the idea here is that our latent space xi is going to be drawn from an independent Gaussian distribution with zero mean and a covariance matrix, just a diagonal matrix. What we're going to assume then is that actually our noiseless observations, so if we're going to take X and we're going to project them onto the higher dimensional space, the space of size n, it's going to be noiseless, and it's going to come from this distribution here. It turns out to be a Gaussian process, where we have a zero mean vector again, which we're going to learn in the posterior is going to be something quite different. And then we have a kernel. And that kernel is going to be the mathematical component that implies smoothness. And so actually we use a bunch of different kernels because it turns out that the different dimensions that we estimate in this space actually have quite different levels of smoothness. So we have a very smooth kernel, something called a square exponential. And then we have three other kernels that are based on Matern kernels, which are progressively less smooth. And so we can still differentiate them. We can still get the inverse of this function, but we absolutely impose these variabilities. Just so again, I'll show you some examples of kernels that we estimate that really describe specific dimensions nicely. So the thing that we do differently from the standard GPLVM, all that is pretty standard so far. The thing that we do differently, specific to single-cell RNA sequencing, is we add this heavy-tailed residual. So what that means is that basically when we think about projecting this noiseless observation, this f of x, up to the actual true observation, y, instead of having a Gaussian residual, which is fairly normal, or which is fairly standard, I should say, we have a student t-distribution. And what that means is that actually these points can lie fairly far from the actual manifold itself without actually drawing the manifold away from where it should be. So in other words, we can have outliers in this space. We're not going to assume a Gaussian distribution around the manifold, which is absolutely not warranted for the kind of data that we're looking at. So this is a heavy-tailed residual, and it lets us handle outliers without warping the space. Okay, so that's the basic model. Let's talk about why we actually use this. So here's a really cartoony version of why we do this, although I have to say these are real data. Real data in the sense, real simulated data. So it's not just a drawing. So here's the red points on this simulated manifold. We see the manifold looks something like a U shape, a sort of a part of a cylinder. And the red points are generated as points from that manifold with Gaussian error, or actually t-distributed error in this case. If we just estimated the model with the normal error that I showed you above, we would get something like this middle figure, where it's just a mess, where you see that the outliers, the points that are far off of this manifold, actually drive the manifold to these crazy places that they really shouldn't be. This is a function of the fact that we're using squared error in the normal model. And it's totally inappropriate for a lot of the data sets that we've seen for single cell data. If we do the t-distributed error, however, we're able to be robust to these points that lie way off of the manifold. And you see we learn this really nice smooth manifold that looks a lot like the simulated manifold from the leftmost figure. So again, when we think about why we want robustness, these Gaussian residuals are going to force this mean function to be skewed away from the true mean function, just because, again, of the squared error that's implied by a Gaussian. We use a heavy-tailed residual to be robust to outliers. And then we find that this really smooth space that we're able to estimate now from this heavy-tailed distribution allows us to do a really nice job in downstream analyses because we have this really smooth manifold on which to work. There's actually no additional parameters. So there's one additional parameter, I should say, is degrees of freedom for the student t-distribution. And we actually just follow prior work from Neil Lawrence and colleagues that basically say if you set it to 4 everywhere, it kind of works well. And we've tried a bunch of different things, and actually 4 seems to be kind of a magic number here, so we've been doing that. I agree that we should probably think more about that downstream, but right now we're going to stick with that. The inference is actually quite easy. We actually just do the same type of Gaussian process latent variable modeling that we can do. It's a stochastic variational inference, and it works quite nicely, and it's very robust as starting point. Okay, so Once we build that that manifold, we can do things on top of it, like I said. So the first thing we did was just do k-means clustering in these latent dimensions. So here you're seeing the points. Each of these are colored by the cell type. So this is the Pollen et al. Data set, which is about 301 human cerebral cortex cells. And what you can see here is the three latent dimensions of this data set for a GPLVM. And the colors represent the different cell types that exist in this data set. These are pre-labeled, although we didn't include the labels when we did the clustering. And you can see that in this GPLVM, it does a nice job of separating these clusters. So we can compare that to a bunch of different approaches. So here's our tGPLVM clusters. Here's the zero inflated factor analysis that was work put out, I think, in 2014. And it basically assumes a zero inflation on a Gaussian latent variable model. It's a linear model. And then the last one is PCA, where we can, again, do principal components analysis and just take the first three PCs and plot each of the cells in each of the spaces. So visualizing this, you can see that tGPLVM does a nice job relative to the others, although I'm not going to say it's like a total slam dunk here. But one nice thing that you see is that there's this particular set of cell types, these yellow and brown cells here that are kind of hard to separate in ZIFA and PCA, whereas tGPLVM does a nice job of separating those. So another thing that we can look at is as you scale up in the number of latent dimensions from 2, which is sort of the naive number to choose, or the number that you choose if you were just visualizing the data naively, to 9, where actually you're probably pulling out some really interesting variation amongst the cells now. You can see the following performance. So this is adjusted RAND index on the left, and the right is NMI, normalized mutual information. And the idea is the higher for each of these scores, the better we're doing in terms of clustering by cell types without being aware of cell types. And so a couple things to notice. tGPLVM does fairly well here, but I wouldn't say, again, it's like a complete win over ZIFA, although I think it is something of a win over PCA. The reason that t-SNE is just sort of these scores degrade very quickly in t-SNE, and there are sort of mathematical reasons why this happens, having to do with peculiarities of high-dimensional multivariate Gaussians. And so you can see, again, the score degrades pretty quickly. So what this means to me is not necessarily that we're doing the best, but that one thing that we should look at when we think about these low-dimensional models is the ability to actually learn higher dimensional, low-dimensional models, in the sense that when we think about each of these dimensions and what they represent, we'd like to pull apart and tease apart as much variation as possible. And the ability to scale up from 2 dimensions to 20 dimensions or 100 dimensions actually makes a big difference in terms of the downstream data analysis that you can do. So I would argue that it's really important to select among the methods that seem to scale well as the number of latent dimensions goes up. So another thing we did was to look at the cell cycle labeled, or cell cycle stage labeled human embryonic stem cell data set from SMARC-C. So this was a data set that was published in 2015. And these are unsynchronized cells. They're oscillating in cell state. So these are XYZ for our particular latent manifold. And you can see that the developmental axis is actually the Z-axis, and this is going to be colored. It's not really colored by the developmental stage. Oh, sorry, the manifold is colored by the developmental stage. I'm sorry. So what this means basically is that the Z-axis of the manifold basically perfectly correlates with the marker of which cell stage we are in. And so there are 2 things to notice here that I think are interesting. First of all, this is a cylinder. So the smoothness of this manifold has let us actually identify a cylinder, which is really cool because we're thinking about basically how cell cycle is in itself a cycle, and cell stage is going to be some kind of trajectory in this space. And you can think about these developmental trajectories being not just lines, but actually more like straws, where the cells actually cycle through cell state and move forward in this developmental trajectory. And that's recapitulated here. So the second thing to notice is that all of the yellow points on this visualization are ones that are way off the manifold. So if we hadn't used that t-distributed error, these yellow points would be pulling the manifold way away from this sort of cylindrical design, and we wouldn't have been able to recover a cylinder for this space. So that's kind of a win for this robustness. So another thing we can do is look at how representative the latent dimensions are with respect to important genes or important features of the cells that we are interested in looking at after the fact. So here what we've done is taken this CD34 positive cells from 10x sequencing and done tGPLVM to map them to a three-dimensional space here. And here's a visualization of that space. And if you remember, of course, our original space is actually 20,000 genes. And we've mapped it down to 3 dimensions. So now we have a three-dimensional space. But if you overlay expression for this particular protein, what you're going to see is that there's a really nice gradient of this protein in this three-dimensional space. And again, what we're doing here is ideally identifying gradients, sort of signals in the data, patterns in the data that we can then go ahead and query using things like highlighting specific proteins or gene expression levels within those cells across the different dimensions to try and sort of put intuitive labels on what exactly each of those dimensions represent. So another thing we can do naively is to take our latent dimension, our latent manifold. So here's dimension 1 and dimension 2 for this data set from Lomberg et al. From 2017. And so these are 409 plasmodium infected cells measured over 7 days. So you can see the day of the measurement taken either 0, 2, 3, 4, or 7. And the idea is to watch how these cells essentially develop over this time. And so what you can see here is that in general, first of all, there is this nice circle representing each of the different phases of the trajectory across time. But then on top of this, we can do a really simple thing. We can put a nearest neighbor spanning tree, and here you can see what we find from this in this low-dimensional space, where there's this. Here's the root right here, the star, and here's actually I forget. I'm not entirely sure what the second star is, but this is the root of the tree. And you can see that the nice thing about this is it basically very nicely separates the 0 from the 7th day. A lot of the other methods that we looked at actually separated it, on the 4th day or the 3rd day. And second of all, you get a really nice description of how each of these cells relate to each other. You can see a little bit of clustering over here, and I'm not entirely sure what that is. It seems like that space for some reason didn't get sort of expanded enough in the tGPLVM, but regardless, we see a really nice spanning tree being developed on top of this model. So taking these ideas, the next thing that we really wanted to do was use our model for batch correction. And so the idea, again, is that if we have a handful of single-cell RNA sequencing data sets from a bunch of different individuals from a population, where we've actually also measured genetic variation, for example, or other types of covariates, exposure to a drug, for example, time series data, we'd like to understand variation across that covariate. That accounts for batch, because batch in general confounds these studies pretty drastically. So we wanted to use tGPLVM to be able to do this. And so the idea there, exactly as Rahul was saying, is that we'd like to perform some kind of manifold alignment that allows us to control for batch effects. And so we had, going into this, we had sort of 4 desiderata, if you will. These are the behavior that we want to encode in our method for this particular space. So in particular, we'd like a mapping between high and low dimensional spaces that allows us to remove variation due to batch. So what this basically means is if we have a point in the low dimensional space, we'd like to be able to map it to the high dimensional space and vice versa. So essentially what that corresponds to is the function of x that we described earlier. And we'd like to be able to do the inverse of that, right? So the function inverse of y, that'll be able to map us back to x. So we have a 2-way mapping in the low and high dimensional space. The second thing we'd like to do is to actually estimate uncertainty. And again, the model that we have is a probabilistic model. So for any single point in this space, we're able to estimate uncertainty and give sort of confidence intervals because we're estimating a full posterior distribution. The next thing that we really wanted was to allow reference-free regularization. And in particular, we want to preserve the variation in this regularization from all the sources that we know except for batch. Batch is the only type of variation that we'd like to remove. Of course, if there's variation that's correlated with batch, we may remove that as well, but that's a bigger problem. If the variation is not correlated with batch, then the goal is, again, to leave that variation in here and not regularize that. And reference-free basically means that we're going to make the assumption that we have no ground truth. There's not going to be any samples that we get that do not have perturbations to the sort of ground truth from batch. So everything is perturbed in this space. Finally, we wanted to make sure that there was robust alignment even when large portions of the subspaces are not shared. And so actually we can go to the total extreme where actually none of the subspaces are shared in the model that we have, which I'll tell you about. But we can also have the small overlapping groups like Rahul was talking about as well. So the actual motivation behind these ideas came from a project that I'm doing with Jonathan Pillow and Anshi Wu here at Princeton. They're in the Neuroscience Institute. And the idea is actually to take a GPLVM, like we were talking about before. This isn't T-distributed, so it has normal Gaussian residuals. And the idea is to use the three dimension of voxel space that are known, X, Y, Z coordinates in the brain. But then add a handful of other latent dimensions. And so what you can see is that basically when we do this, the original voxel space, where we're just estimating this manifold in the high dimensional space, looks something like this on the left. If we just do a linear projection and basically only think about smoothness among local voxels, we get something like this. And you can see that the covariance matrix plotted below, the heat map of the covariance matrix plotted below, loses a lot of information. Sorry, is there a question? Okay, good. Finally, if we actually use our brain kernel, is what we're calling it, this set of fixed effects X, Y, Z, plus a bunch of latent variables that we estimate from the data, we see basically a really nice reconstruction of the covariance matrix. And what you can see from the actual manifold that we learn is that we get a couple things that we know about the brain. For example, we get bilateral symmetry of different portions of the brain. We also get pretty distinct cutoffs between very local regions. So when you go from one region of the brain to the other, even though they're adjacent in the actual brain, we actually see that they get split pretty dramatically within our brain kernel, just because there's very little information, signals passed from one to the other. So this gave us hope that we could do something in the same vein with RNA sequencing. So what we did was essentially build a semi-supervised version of the tGPLVM model that we had. So the idea is essentially we just have a single fixed covariate, and that's going to be batch. I should say that we sort of did this arbitrarily if we wanted to control for anything else, whether it's genotype or exposure or pretty much anything else, population. We could also include that as a fixed covariate too. So this is just kind of arbitrary proof of principle. So it's semi-supervised in that we have one fixed covariate in this case, and we have a large number of latent variables. In this case, I think we actually fit it with 9 for most of the data sets that I'll show you. So the latent dimensions, again, are going to represent some kind of shared space across batches. It's possible, again, that you could actually have entirely distinct spaces across the different batches, but we're going to make the assumption. Actually, we're not even making that assumption. It would be nice in terms of recovering signal if there was some shared latent space, but it's not necessary. So the nice thing about this model is that we're basically doing exactly what we wanted to, which is doing manifold alignment across these different batches. So now we have the variation in this latent space, and all the variation that's been removed by modeling the separate batches is going to be controlled for, and we can toss it out because it's modeled explicitly. And then finally, the idea now is to perform all of our downstream tasks on this shared latent subspace. Or we can do another thing, which is take the cells that are mapped in this lower dimensional space and map them back to high dimensions where now we've actually controlled for batch already. So those are our goals here. So I'm going to talk to you about the exact same data set that Rahul was talking about, which is this pancreas data set. Well, it's actually 4 data sets, I should say. They were performed from 4 different groups and 4 different methods. I only included one of them here, but the 4 papers are extremely cool to put the data together, as Rahul did. The idea here is that we're going to color the cells by the ascertainment technology. So here is, on the left is tGPLVM, where we haven't put in batch. So we're just modeling the 2 latent dimensions without including batch. And you can see this sort of scary, dangerous thing where there's really nice separation of each of these cells by which technology they were ascertained with. If we use the tGPLVM, where we actually have this fixed effect that's batched, we see the following, which is there's this really nice description of each of the cells where all of the different cell types are essentially, all the different technologies that they were ascertained from are intermingled in this lower dimensional space. And so with this kind of model, actually, all of the 4 things that we were hoping to get from this space are satisfied. So here's that exact same projection to latent space where we've colored each of the cells by the cell type, where we did the inference of the cell type ourselves. And so there's 14 different cell types, and you can see again that there's really nice clustering of these cell types in this lower dimensional space where we don't see that the different technologies have sort of perturbed the clusters in a way that's unacceptable. So in particular, when you think about this low dimensional space, we are able, of course, because we have a nice function inversion, to be able to map back to the high dimensional space, where we can map back in a way that actually removes the batch effects. We have really nice uncertainty estimates simply because this is a posterior estimate of a manifold, so we have error bars on this. This is reference-free regularization, so we don't need to have any information about relationships among cells in the original data set, where, again, we preserve variation. If the variation is uncorrelated with batch, we're going to find it in this setting. And then finally, again, we can have a situation where some of these cell types, in fact, essentially all of the cell types do not need to be shared across the batches. And we can still see them in this lower dimensional space correcting for batch. And so I should say these data are really hot off the press. I literally got them 2 days ago. And so now we have a bunch of work that we're trying to do. So in particular, we're now looking to validate these types of models using differential expression and population variation, just to make sure that we haven't removed population variation or variation across different conditions by doing this batch correction. So in conclusion, we developed this nonlinear robust latent variable model to model RNA sequencing in a low dimensional representation. We can do a lot of different things on top of this model, including clustering, developmental trajectory inference, imputation, and visualization. We extend this model to a semi-supervised form of alignment to allow batch correction. And then there's, as you know, as we all know, there's many different ways to think about latent manifolds for RNA sequencing. The reason that we like our approach, and again, it's very, very taste specific, is because it allows us to propagate uncertainty to downstream tasks, which I think is really important to do. And also it allows robustness to outliers, which is a really big problem for a lot of the analyses that are happening. I should say that our group is doing a bunch of work that sort of builds on this that I haven't talked about, including, like I alluded to, experimental design work, where we have 2 manuscripts on that already. Thinking about observations that are not missing at random in terms of dropout. This is a big issue. Combining spatial and temporal data, so not just thinking about these as static observations, but thinking about them in both space and time. And then I think most relevant and also related to the work that Rahul was talking about with the spatial modalities is combining the disassociated RNA sequencing data with other data modalities like spatial data to be able to understand the mapping between those two. And I'd like to acknowledge my awesome group. I have to say most of this work was done by Archit Verma, he's right here, with help from other people and also my funders. Thank you.